Hey guys, so I wanted to talk to you today just a little bit about protein synthesis. And when you look at this process, it's super complicated. And I'm going to teach it to you in just kind of this really general schematic that will help you just kind of get the idea of what's happening. So before I kind of go through this scheme that I have on the board, I want to talk to you a little bit about bringing us back to cells. And we've already learned about that in how the cell nucleus, for example, holds a structure called DNA. And DNA is in my mind, your coding molecule. Everything comes back to DNA. So I have DNA, this code, so to speak, tells me what color of hair I have and how tall I'm going to be and whether or not I'm going to have a certain color skin. All these things are coming down to DNA. And the same thing happens here when I look at protein synthesis. This DNA is going to be what codes for you every protein in your system. So you're looking at structural proteins. You're talking about proteins that allow for enzymes to work and allow for all these different things to happen inside of your system. And that's what I want to teach you about today is how does that happen? So first things first, DNA is found in our nucleus. Okay. Now DNA, which is going to be this double helix code that you see here in black, I'm going to tell you more about that in just a minute. DNA is going to produce for you a code called RNA or mRNA shown here in blue. And that code is going to actually leave the nucleus. That code goes out. And if you remember the organelle that's responsible for allowing for proteins to be made, that's called the ribosome. And this code right here goes out to a ribosome. And that ribosome helps you then produce this other type of code called tRNA, which brings in then a protein. And that formation, and it's kind of how you get it. It's really just a bunch of lettering. It's a lettering system that starts here. We break that down, we get this code, and then this code becomes this code, and then this code becomes this code. And this down here at the bottom here in black is going to be the protein sequence that gives us function. Now, I'm going to teach you about how you do this because I'm going to actually have you do an activity in formulating this process for me today. But here's what I want to be sure I tell you is that this is where you begin. This code up here is a bunch of what we would call different types of chemicals that are allowing for you, nucleotides, that are allowing for you a code of some kind. That code means something. When you talk about this code, it brings about a protein sequence, okay, that gives you functionality. This code determines functionality. And I want to mention to you that proteins, whatever sequence this has, okay, will create a certain folding pattern. And that folding pattern brings about function. And this is kind of where we come into different diseases and we come into different problems in the human body because one single letter, okay, that's mutated or broken down or not correct or just deleted, for example, can yield for you a completely different code down here that gives you a completely different function. Many cancers begin this way, where I've mutated something. UV rays, for example, can delete some of this code, and when we delete this code, then I get a different protein sequence, and that's how we yield then cancer and different problems inside of the system. It all comes down to what this code says, and when there are problems associated with this code, I get problems in my protein. And if I get problems in what this protein says, then it's not folded the way it's supposed to be. It's kind of like trying to do origami, right? You have this piece of paper and you have these instructions, and that's exactly how it's going to go. But if you fold that thing one way in the wrong direction, then your end result isn't what it's supposed to be. The same thing happens here. If I lose one single piece of this code, then I get a whole different sequence at the bottom. So first things first today, and I'm going to have you do this in an activity. You can see here that I have my 4 original bases associated with DNA. These 4 bases, as you may already know, adenine, thymine, cytosine, guanine, A's, C's, T's, and G's. Now, A's and T's match up together in DNA, and C's and G's match up together in DNA. So if I give you a code of DNA, you can see up top here, I have A, T, G, A, blah, 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 all the way down. So going with this code up here, if I just give you this top code, you should be able to tell me what matches up with A, and that should be a T. What matches up with T, that should be an A. And so you get this kind of double sequence code where each of these guys are paired up together, okay? So I have all of that code written out for you here, and I would have you guys draw this out, push pause, and draw this out for yourself. Now, so this is DNA code. Once DNA comes into play, we want to make a protein out of it. We have to unzip this, and that would be what would happen. An enzyme comes in and tears these 2 strands apart. And one of these strands, and I just chose this bottom one, it's called the parent strand of DNA. This parent strand of DNA then is going to form a molecule of mRNA. Now, this messenger mRNA is going to be a complementary code of the DNA parent strand, or what I began with. But here's the kicker. In RNA, you no longer have T's, okay? You have A or U's now. And a U is uracil. So now I'm going to add the base uracil. Uracil, uracil. And so everywhere I have a T, I have an A, because I already know that. Everywhere I have an A, I no longer have T's, now I have U's. So you can see how I've matched these up once again. I've created a complementary strand based upon what I should have. Okay, so I have all those matched up. Now, this is what you'll have to do. You'll have to create a code for me. Okay, it's really simple. Just remember that in RNA, you don't have T's, you have U's where T's should be. Okay, so everywhere basically that I had, in this case, an A, I have a U now. At this point, I have an mRNA code, and this code is the one that would go to the ribosome. Okay, this one is going to go out to the ribosome and allow for us to produce then a protein. Now, here's what's kind of interesting about it is I have this big molecule, and the strand comes through, okay, and it hooks up to this big kind of circular ribosome. And as it goes through, it's kind of like an assembly line in a way. And this code that you have is going to be split into threes now. And these threes are going to match up with something that we call a codon. And this in green is called a codon, and it's also known as tRNA. And this tRNA, or transfer RNA, is going to come in, and it's going to bring in a protein. So what you have is a complementary strand. So look, if I had AUG for my mRNA, my tRNA would be the opposite. So it should be a U, an A, and a C, right? Because of my matching system. So everything comes back to what it matches with. A's with U's, right? And then C's with G's all the time. And so when I look at this, this is what we're getting. And so in this case, I know you don't know what proteins are. I'm going to give you a list of how to use that. There's a graphic that you can use. It's real simple. In this case, this transfer RNA brings in methionine, and this is going to be the protein amino acid that we're talking about. So this codon matches up here. This codon is serine. This one is phenylalanine, and this one is a stop codon. Now, I didn't go long. These things will go for miles, okay? But what I want you to see is that this matching system, this tRNA, always brings in one of these amino acids. And these amino acids will eventually bring in what we call the stop codon. And this tells it to stop. Okay, we're done creating this protein at this point in time. What we're going to do is have the end of that protein. So this strand on here is known as a polypeptide. It's your protein strand. And this guy right here, depending on what amino acids are in what sequence, will depend upon how it folds together. And that will give it functionality. I would mention to you as well here that this process can be quite lengthy. You can have some proteins that are thousands upon thousands of amino acids long. There's potential that some of you have heard that amino acids are the building blocks of life, and that's totally true here. I'm building up the protein, your life, okay, by putting together these amino acids. And these amino acids come back to initially DNA, which created mRNA, then tRNA, and then gives me then a protein sequence. So be prepared. I'm going to give you some instructions on how to do this on your own, but this is basically protein synthesis. So you just coded out a protein for yourself, and we're able then to determine how that protein would fold. Don't forget, if you have a mutation, if you delete something up here, something goes wrong, you get a whole different sequence down here. If you get a whole different sequence, that changes the folding pattern. Therefore, you potentially have misfunction or a disease of some kind that doesn't quite work the way it should. So hope you guys are having a good day. This is Protein Synthesis. Hang tight for an announcement.